Here we have moved round to the assembly test area again, which we saw on our walkthrough. The operator here is in doing the preliminary checks on the console, checking out the printed circuit boards in the system. We have a signal simulator that we actually run some basic tests to make sure that the system as an electronics module works correctly before we link that up with the rest of the system. We've now completed the detailed look at the manufacturing line. I'd like to give you a quick overview of how the process itself works. Basically, the material is drawn to the floor by the operators, and they begin their assembly activities as we've seen. The stations are planned in such a way that each two stations make a cell, and it takes approximately 1 day for us to complete the activities in a work cell. So in this particular line, we're actually seeing like a 5-day event for the material to pass from station 1 through down to station 9. The element in terms of what triggers the operation is that marked out on the floor, you can actually see that there are some black squares. If the square is open, i.e. it doesn't have a cart in it, it means that it is open for an assembly to be passed into that particular cell. Should it be occupied by a cart, then it means no material should move into that particular area. The system is therefore self-pacing based upon a balanced manufacturing line.
Thank you. All right,
Rolling, stand by for
Ray will be the first one to talk.
okay.
Speed. Put it in pause for just a second. I'm sorry.
Rolling, stand by for speed. Speed.
This afternoon we're in one of the research labs at BDIS talking with Dr. Dieter Rechtenwald, Group Research Leader. Dr. Rechtenwald, could you show us how a cell sample is analyzed in the VacStar Plus cell sorter?
Yes, sure. You start out with a sample preparation, and typically you have your cell samples in a small volume in test tubes. And in this case, we have analyzed 3 tubes already, and I'm taking out the 4th tube. I'm mounting it on the instrument. And then when I pressurize the sample containing the cells is introduced into this tubing. And I should mention one other detail. You want to agitate your cell sample because cells are heavier than most buffers used to suspend them. So they slowly settle down, and you will have to resuspend them to introduce them into the instrument. And the cell sample moves into closer to the measurement area, where it is met by another fluid stream, and you can see some other fluid tubing here, which contains so-called sheath fluid. And this fluid serves to focus the center sample stream to narrow it down to a stream which is about 1 cell diameter wide to create a one-dimensional file of cells. And I open up the instrument. When I do that, I will trigger a laser safety lock, and the measurement actually will stop. And here you can see the sample stream now. And this sample stream is intersected by a laser beam about here. And whenever a cell goes through this laser beam, there is a light flash which gives us scatter and fluorescence information. This information is then utilized by the detectors and the data acquisition and analysis system to make decisions on whether this cell is wanted or not for sorting. And we talk about these aspects later. And then if a cell is wanted for sorting, the sample stream is charged through these wires, and you see these essentially condensator plates, and they are charged also. So if you have a charged droplet in this stream, then this droplet is deflected, and you see collection tubes for cell samples here. We can sort at cell rates of up to close to 10,000 per second. And so after several minutes, you have a cell sample which can be analyzed for biochemical parameters, for molecular biology, or other research applications. Another detail I forgot to mention is that this stream is shaken by a piezoelectric vibrator to break it up into little droplets shortly after it leaves this nozzle tip, and then charged droplets are deflected, as explained.
Okay.
Well, your idea.
Rolling, hold speed,
Stand by.
and speed.
Don't say anything. Just do it. Okay, pause the tape. That's good.
Rolling. Hold speed,
Okay, go ahead.
speed.
Get your hand out. Oh, okay, I'm sorry. Do that again and hold it for a little longer. Okay, great.
Rolling.
Good.
Hold
Stand by.
speed.
Stand by.
Speed.
Okay, just hold it for a second. Okay, now hold it. Okay. You stop tape?
Yes.
Or
I
pause?
opened the next. I opened up, and you might want. No, the open up was, I think, was clear enough in the macroscopic picture. So, I think what you
Now, the next thing you said was moves to the
Rolling.
stand by. Don't do it
Stand
yet.
by. And speed.
Okay. Okay. Okay, do that again, and then I didn't realize you were going down. Do that. Just
shall
do
I
the
go
same thing
not?
again.
Shall I go down?
Yes. Okay.
Rolling. Hold speed. Speed.
Okay, stand by. Okay, finger in. Okay, I think I didn't
That
cue
was-
you.
yeah, I wasn't sure.
Yeah, okay.
So you were-
I need to cue you first, I'm sorry, that was my mistake. We're still rolling.
Okay.
All right, stand by. Stand by for your cue, Dieter. Okay, zoom out a little bit. Go ahead, finger in. And finger out, great. All right, pause tape.
Rolling. Stand by for speed. Speed.
Okay, just everyone stand by here. Okay, tilt down. Point. Okay, keep rolling. just go wider, just now, just right now, just widen out on.
Okay.
No, I was thinking of the container area down here.
Okay.
Just,
You ain't
yeah.
with the move?
No. Okay. Good. Pause tape.
Rolling.
Stand by. I'll cue you separately.
Speed.
And remember, you're going to say that beside the cell sorter. Okay. Start to pull out and go.
Right beside the cell sorting measurement station, we have our electronic console, and what we are seeing here is a display of pulses, and this display is generated when cells go through the laser beam in their fluid sample stream. That means when the laser beam hits a cell, you get a light flash, as I had explained earlier. And when you look at the display, you see that the intensity of this light flash measured by a photomultiplier goes up as the cell enters the laser beam area, and then goes down as the cell leaves this area. We have 4 different traces because, as we will see later, we have 4 or more detectors on most modern flow cytometers. So each of these traces corresponds to a different light measurement for a single cell, and each of these pulses corresponds to a single cell going through the beam. And you should note that the time between 2 divisions here is only 5 microseconds. That means a cell spends less than 3 microseconds in the laser beam, and then there is time for the next cell. As you can also see, you don't see any second pulses here. That means there's a lot of spacing between single cells, which is important because you do not want to measure 2 cells at the same time. Okay.
Okay. Now, why don't you carry on? Just describe the other one.
The upper display now is a display further derived from electrical signals. These electrical signals go through a peak and hold circuit, and the height of these 4 signals is measured, and the height of 2 of those signals can be displayed at the same time, and it gives us information about properties of those stained cells, or about their light scatter. And in this case, we plot one color of fluorescence here. You can see here it says fluorescence 2, here it says fluorescence 1, which is another fluorescent color. And each of the dots coming up here and disappearing here corresponds to a single cell going through this laser beam. And on this display now you can see that the cell sample under investigation is composed of 3 major populations, and then you see dots all over the screen, and those dots might indicate some minor populations. And all of this can be done without complicated computer circuitry, but then in modern flow cytometers, this data is sent over to a digital computer for further analysis.
So in essence, what you're looking at in the top screen is the size of the particles of the cell.
It can be the size, and I would have to switch the parameters using these controls. What we do display here in this instant is fluorescence intensity in 2 different colors. And to make it a little more colorful, this is our green channel, this is our red channel. So this would be green fluorescence emission and red fluorescence emission. These cells would have a very low level of fluorescence emission in either color. Here we see predominantly green fluorescence emission. Here we see predominantly red emission, and this area would be for particles. And you see some come up once in a while which have both green and red fluorescence emission. And that's a way you determine subpopulations.
Thank you.
Okay, pause the tape.
Rolling. Hold for speed. Speed.
all you're going to do now is describe just with the point. That's all we're
Yes.
doing this for. Okay, go ahead.
Here we have information extracted from the electrical pulses, and this is information for single cells. And this is green fluorescence intensity, this is red fluorescence intensity. This population is cells which have predominantly green fluorescence. This population shows cells which do not have any substantial fluorescence, and this population shows cells which have predominantly red fluorescence. And in this area you have a few cells which have both red and green fluorescence. And again, I want to remind you, each dot showing up corresponds to the measurement on a single cell.
Very good. Okay.
Rolling. Hold for speed. Speed.
Okay, now when I cue you, go ahead and just point to that and then pull the pointer out. Okay, go ahead. Okay, pause the tape and let's find.
Rolling. Hold. Speed.
Stand by. You're going to go in and point and pull it out. Go ahead. Great.
Now I just noticed it doesn't make any scientific.
Rolling. Hold for speed. Speed.
Okay. And why don't you just explain very briefly what you're doing here, just so we'll all fit together. Go.
Okay, here we see the electrical pulses from our light detectors, and you can see as a cell enters the laser beam, the measured intensity goes up. As it leaves it, the intensity goes back down again.
Okay. Pause. You said something about the 4 different.
Hold for speed. Speed.
Okay, stand by. And that's all you're going to do. No explanation. Just do that. Go. Okay. And now just keep rolling. Can you point to what you did for the spacing in between?
Oh yes.
Go ahead, do it. Okay. Just pause tape.
Rolling. Stand by. And speed.
This electronic console is interfaced with a modern computer workstation for further and more complete data analysis. And you will see the same kind of display again, in this case in real time on the screen of this computer workstation. If you remember, we saw this display from data very close to the analog end, and we reproduce it on the computer, except in this case we have digital information stored in the computer, and you also can see more parameters at the same time. In this case now you see parameters relating to cell size, forward scatter and side scatter, which is done without using optical filters, and again fluorescence 1 and fluorescence 2 as we had seen before, but in the digital domain. And then on a computer workstation you have more flexibility in terms of displays. So in this case you see 3 parameters at the same time. And here you have a display which is used very frequently in flow cytometry and cell sorting, because it also gives you some idea on the relative frequency of cells in cell populations by so-called histogramming. And in this case you see by fluorescence 1 alone we have 2 major populations. If our biochemist would be interested in this population, which is stained with red fluorescent color, a marker could be set around this population on the computer screen. This information could be downloaded into the cell sorter, and the cell sorter could be activated to sort this population for further analysis.
Good. Thank you.
Great.
Okay.
That's a wrap then.